This is unfortunately not eligible, or most of you are not eligible for this. We did that survey right at the very beginning. But if you know someone who's interested in the Build Excedo program, please apply. I can give you some information about it. I have a couple of Build Excedo students who are currently working with me. But it's 3 years remaining in your undergraduate degree. Hopefully none of you are going to be taking 3 more years. It's the plan. And they must be enrolled full-time spring and fall of this particular year. Actually, I think that might be fall 2019 rather than 2018. No, that'll be that is fall 2018. It's the end of this year. Okay, so as usual with my reviews, there's nothing new here. All of this is stuff that we have covered before, and this is stuff which I think I didn't do a very good job describing the first time around. Please ask lots of questions as we go through here. I'll also be covering some of the things that people have asked me questions about via email, almost all of which I've posted to D2L. So if you haven't gone into the discussion section and looked at questions, please go ahead and do that. Hopefully they will be useful for you. There's one exception in terms of the slides, and I'll tell you about that when we actually get there, assuming that we do. Couple of things here, again, just pulling out what I think are particularly important concepts. No guarantee whatsoever that these are going to be things that are going to be on an exam. So the idea really here of the tree of life that I'm not sure I did a very good job explaining the first time around is that all cellular life has a few things that are fundamentally the same about it, like using DNA for its genetic material, like using ribosomes, which are mostly RNA, to make proteins. So if we're interested in fundamentals, which is really mostly what we're talking about in this class, it's very important to think about things that are conserved, i.e. that are present in lots of different organisms of a particular group. The bush of life basically just says there's not a nice linear relationship between all of these different things. And the, sort of the whole big deal here is that, microbes really rule. This is we're on a microbial planet. Yes, we may think that we're the most important, understandable, but basically it's microbes. Microbes have been here since way before we were, and will be here long after we're gone. Conservation. We've talked about conservation just as present. That particular process is present in lots of different kinds of organisms. But we're also talking about sequence conservation, because sequence, and when I say sequence, that could be amino acid sequence, it could be nucleotide sequence. If you find things that are similar in those sequences, that means that they're conserved, but why are they conserved? Life has been evolving on this planet for plus or minus a few 100 million years. Okay, we're a minuscule portion of that, of course. This is about 4 billion years. And so any sequences which are the same in multiple different organisms are going to be important in some way for the function of whatever that molecule is. So if you find something like this is the ribosomal RNA gene, where we've got sequences in green which are very well conserved, i.e. they're extremely similar between humans, and this is just an example of eukaryotes. Turns out eukaryotic sequences are almost completely identical here. Methanococcus, which is an archaea, on the second line, and E. coli, a bacterium, on the third line. So in that green box, these things are practically identical. And that means that over 4 billion years of evolution, these sequences are really important. On the other hand, what's in the red box is quite different between these different sequences. And that means that it's probably not as fundamentally important for all organisms, but could, of course, be important for those individual organisms and maybe how they're different from each other. But again, the key here for conservation is thinking about something is present in a particular sequence, in a particular structure. It's there because of evolution. So hopefully that makes more sense. I think I did a pretty crummy job of explaining this again the first time around. One of the things that you can learn through this, if you just look at the minimum genome complement and say, hey, this is the only thing that's are present in this particular genome, which of those are the most important for a cellular organism? And the take-home message here is it's really about proteins. If you look at the translation and amino acid metabolism, the very few number of open reading frames, we'll talk more about open reading frames later, but basically you can think of these as genes encoding proteins, are mostly important for translation and amino acid metabolism. So translation seems to be really key for all cellular organisms, which is the big take-home message from this one right here. So how do you get these various different genes through the evolutionary process? They're basically two main mechanisms, giving you orthologs and paralogs. Orthologs are those that happen through evolution, giving you different species. So these are the kinds of things that you would see, again, conserved over very large distances on those trees, which are just literally number of changes in sequence between any two different organisms. So that's where you get these particular divergence in those genes, but then also conservation for a particular function. On the other hand, just to confuse things a little bit, we don't have 182 protein coding genes in our genome. We've got about 20,000 of them. So how do we go from something, in fact, the 182 is probably a reduction, but around 500 genes probably, protein coding genes, to the 20,000 that we currently have. A lot of this has to do, with gene duplication, and play this instead. Gene duplication, which is happening in a particular organism. This can be a whole genome. People ask me about plants. Often genomes have whole genome duplications that take place. Or it could be just a gene duplication, which happens, usually having to do with recombination happening in meiosis. So now you end up with two copies of a particular gene. The two copies of those particular genes are paralogs. There's one other way that you get new genes. What's that? Horizontal or lateral gene transfer. What does it? The most important organisms on the planet? Yes, the viruses, exactly. There are other ways that you have horizontal gene transfer as well, and we'll talk about those a little bit later on. But viruses are probably one of the most important. Lots of people have asked me about, Dr. Stedman, what are we supposed to know about all these model organisms? A couple of things about model organisms. Again, getting back to conservation and evolution. Basically, these model organisms are organisms that it's relatively straightforward to do experiments on, or in the case of the bottom one here, to work with in terms of doing individual experiments. So if we talk about bacteria like DNA polymerase I, DNA polymerase III, etc., all of this comes from the study of E. coli. So when I say bacteria, 99+% of the time it's going to be E. coli. Why? E. coli is easy to grow, there's good genetics, and it's a relatively small genome. When we talk about archaea, most of the time that's going to be studies that have been done on Sulfolobus sulfatericus, because that's what we grow in my lab, but it's also a quite well-studied system. You can't study all organisms because there aren't enough people on the planet for each of them to study all of those. If you think about eukaryotic model organisms, there are a couple of different ones that are being used in different ways to answer different questions. If you want a really simple, very small eukaryote that has a lot of the very basic conserved mechanisms, sequences, etc., for understanding eukaryotic biology, Saccharomyces cerevisiae is great because it's a small genome, easy to work with, great genetics. On the other hand, it's a single cell. So if you're interested in something that's a little bit larger, you're interested in cell biology, development Saccharomyces cerevisiae might not be the best model system to study. C. elegans is great for studying cell biology because you can see all of the cells which are moving around in it. However, if you are interested in plants and chloroplasts, C. elegans is probably not a really good system to study. Arabidopsis is a really good system to study. And again, these are things that people have developed over the years. If you're interested in general development, particularly in terms of genetics of, again, some very fundamental aspects of eukaryotic molecular biology, Drosophila is a great model system. We're talking a lot about Drosophila so far a little bit. We'll talk more and more as we move on through the course. But none of these things have backbones. Okay, if you're interested in something with a backbone, zebrafish are a really quite good model organism to use for this. That's great, but okay, if you're interested in something that's specific to mammals, say an immune system, particularly adaptive immunity, then you might want to study something which is a mammal and something that you can do a lot of experiments on. You can breed the way that you want it to breed as opposed to that last thing on the list here. So basically the idea here is that you pick a model organism to study a particular process with that makes the most sense, what has already been done in those organisms, etc. So a lot of this has to do, again, with genetics and sizes of genomes. If you don't have a good genetic system, it makes working in a particular organism very, very challenging. Okay, so why are these things so useful? And again, the classic example here is the baby and the mouse, which have exactly the same mutation in their genomes and give you exactly the same phenotype. This one's a lot easier to study than this particular one. The further back in time that you go, unfortunately we have a fossil record that we can compare and know how old each of these particular organisms are, and then through sequence comparison we can compare, in this case, humans and fish, and this would be the vertebrate fish. There are also some of the cartilaginous fish which are even more deeply divergent here. But again, it's the whole idea is conservation and evolution. You know, evolutionarily, we're pretty darn similar to mice. Evolutionarily, we're pretty darn different relative to E. coli. Nonetheless, there are a lot of things that we can learn. Again, E. coli and us, if it's true for the E. coli, it's true for the elephant, then some of those things we can learn studying E. coli, they'll be directly applicable to us. Some of the things we learn in E. coli, we'll talk about this again later on, turn out to be not at all applicable for us. So any questions sort of on that genome comparison kinds of things? Otherwise, I'll move on and do our quick chemistry review. Again, probably a year worth of chemistry in 5 minutes. Important things here are really that we have covalent bonds, which are very high energy, and lots of non-covalent bonds, which are relatively low energy. In terms of covalent bonds, the ones that we're going to talk about in this class basically are going to be any kind of nucleotides and how those nucleotides are put together in terms of DNA and RNA. We're talking about covalent bonds that are peptide bonds formed between amino acids, a few covalent changes which happen to proteins, a little bit later on, nucleotides as well, phosphorylation, for instance, ubiquitination. All of these are these high energy covalent bonds. How do you get those? In terms of formation, again, energetically really hard to make because they're very high energy. You have to then couple these to other reactions and hydrolysis reactions coupled to these condensation reactions. Forming these covalent bonds is always going to be a condensation reaction as far as we're concerned in this course. So those, on the other hand, all of these other weak interactions or other different kinds of bonds which are forming, this we're going to see again and again and again and again and again because there are lots and lots of these. This is because biology is dynamic. If it weren't dynamic, you wouldn't be listening to me and I wouldn't be talking to you. So all of this dynamism has to do with these weak interactions. And we'll spend lots of time talking about the different weak interactions. Classic example that we've talked about quite a bit so far has to do with histones interacting with the DNA in nucleosomes. So lots of different kinds of interactions. They could be hydrogen bonds, they could be ionic interactions, they could be hydrophobic interactions, etc. All of these weak bonding interactions between different macromolecules. So again, most of this course is going to be concerned with that. Really quickly, acids and bases. Acids are those which are giving up hydrogen bonds, hydrogen atoms, I should say, not hydrogen bonds. Those are giving up hydrogen atoms, and then bases are those that are giving up hydroxyl groups. And so what's most important as far as that we're concerned for that? Two kinds of acids and bases. Aspartic acid, glutamic acid, and lysine and arginine. Exactly. So those are going to have stable charges in biological systems, and so they'll be interacting with each other. I mentioned this before, but it bears repeating, and I also think I did a crummy job explaining it the first time around, having to do with the whole idea of a coupled reaction. So these coupled reactions basically means that you can do something which is energetically unfavorable, i.e. forming a covalent bond, if you can somehow couple it to highly energetically favorable reactions. And we're not going to get into a whole discussion of delta G's here. That I consider is much more appropriate for chemistry courses, biochemistry courses, etc. But the idea here, we have to have something which has a larger change in the free energy, which is going to be a spontaneous reaction that is then coupled through this little cogwheel, which is usually an enzyme, sometimes a high energy intermediate, which will then allow a otherwise unfavorable reaction to take place. The magnitude of this reaction, or this set of reactions, always has to be bigger than this one. Okay, questions on chemistry? Wow, it's like 2 minutes of chemistry. Good, happy? Can forget all your Diels-Alder reactions, etc. Yeah, in the back. Hydroxyl groups? It's just the OH ion. So, pardon? Acids and bases. So the base is then when you're losing the hydroxyl. Sorry. Probably speaking too quickly. Yes, down here. Yeah.
Oh, I'm sorry. Your point is
No, that's all right. There are a lot of people here.
the activated intermediate.
Yes.
Are you going to? I didn't know if you had a slide on that. I'm just a little confused about summing up and being able to define that sort of a system.
Okay, so the question basically has to do with activated intermediates, which I'm not planning. Didn't have an extra slide on here. Sorry about that. But again, I'm not quite sure I understand your question, so correct me if I haven't. But basically, what are we thinking about when we think about an activated intermediate? So the activated intermediate is basically going to be the process, maybe we should back up here, of doing some of this coupling. So an activated intermediate, particularly when we're talking about polymerization. So when we talk about polymerization, we're going to have the activated intermediate, which is the nucleotide, I should say the deoxynucleotide triphosphate, in terms of doing DNA polymerization. So normally you'll have, for instance, let's take a good example, thymidine monophosphate. Thymidine monophosphate has to become a thymidine triphosphate before it can get incorporated into the DNA. So that's the idea of the activated intermediate, because, again, you've got to have a larger spontaneous reaction that happens here than one which is not going to be spontaneous, and clearly polymerization is ordering things, and generally you're going to be increasing disorder. Does that make more sense?
Yeah, the example that was given in the slides from that lecture was taking glutamic acid.
Mm-hmm. Yeah.
And it was just kind of
Yeah, so again, to follow up here again so people can hear it, the example in the textbook and the one that we used was from going from glutamic acid to glutamine, and sort of why do that. Just as sort of, it's supposed to be a very general case. And again, if you're interested in biochemistry and all those kinds of things, also exactly the same concept. But as far as this class is concerned, the only things we're really going to be very interested in are going to be those specific ones for polymerization. Yeah? Do I want you to remember each of the delta G's for the individual bond energies? I can't remember them, so I can't expect you to remember them. Now, covalent bonds are much, much higher, about 100 times more than any of those individual ones. I think that's perfectly reasonable. But yeah, the thing is, it depends exactly on the conditions as well. So if you remember one condition, it's not even going to be that applicable in other conditions as well. Okay. You're all doing really well on your chemistry exams, right? Good. Okay, so protein structure. This also seems to be an area of some confusion. And that's because molecular biologists and biochemists use different terms for different things, and I apparently do as well. So 4 different levels of protein structure: primary, secondary, tertiary, and quaternary. So primary structure, very rarely will I use that term, and it's very rarely used. Much more common is primary sequence. And so it's literally what the amino acids are going from amino terminus to C terminus. So that's going to be your primary sequence. That's something which is relatively easy to determine if you're just looking at proteins. It's really easy to determine if you know the genetic code and determine the sequence of your individual nucleotides. So that's the easy part. The secondary structures are where each of the parts of those individual amino acids are going to be coming together. And this is something which is universal for almost all of the amino acids. Darn biology, there's always these exceptions. Proline's that one exception. But here, it's the backbone atoms. So in your peptide, which starts in an amino terminus and ends at a carboxy terminus, you're going to have peptide bonds, which are these amino groups and carboxy groups interacting with each other with various side chains hanging off of them. But these secondary structures are universal because they're independent of the side chains. Because they're all going to be these backbone, i.e. not side chains of the individual amino acids that are associated with each other. And the 2 that we've talked about, which are the 2 most common ones, are the alpha helix and the beta sheet that we'll look at in just a second here. Those secondary structures then associate with each other in tertiary structures. That's where you have your specificity of each of those individual side chains that are coming off of the individual amino acids. So that's how you end up with your tertiary structure. It's the interactions of side chains after you have these secondary structures, which are what's happening in the backbone. In some cases, these tertiary structures will come together as quaternary structures. Quaternary structures here. Now, this is where I think some of the terminology comes in. Polypeptides. What the heck is a polypeptide? So the polypeptide is a stretch of amino acids kind of arbitrarily defined as being more than about 20 of them together. And if it's shorter than that, it's an oligopeptide. So polypeptide, relatively long. So you can talk about a protein as being a polypeptide. The problem is that many people will also talk about this, the quaternary structure of a protein, say for instance hemoglobin, as being a protein, but it's made up of 4 peptides because each of those has an amino terminus and a carboxy terminus. So your protein is made up of 4 polypeptides. And often people will talk about each of those polypeptides as being a subunit of a protein. Okay, does that make a little more sense? Yeah.
Is polypeptide synonymous with multimer?
Okay, so the polypeptide is the individual one N-terminus, one C-terminus. So it's multiple amino acids together. So the multimer will be multiple polypeptides. A protein is often a multimer if it's got multiple polypeptides. And so part of the problem is the poly. So this polypeptide is just that it's multiple amino acids. It has nothing to do with the number of individual sets of polypeptides that come together. Yeah, Damon in the back?
So on the screen up there,
yeah.
it's an unfolded polypeptide with the colors coming off of it. Is that equivalent to the primary sequence that's on the left?
Yeah, so just to repeat this. Yeah, so the unfolded polypeptide, this piece right here, that's equivalent to this right here. So that's going to be a primary structure, primary sequence. There's another question down here. Okay, so this is, okay, I'm going to paraphrase your question, pardon me, which is, what's a domain? And so a domain is going to be part of a single polypeptide that forms a structure. So it could be your whole polypeptide, which forms a structure. It could be different parts of that polypeptide that form a structure. But the domain is always going to be part of one polypeptide. The definition of a multimer is going to be multiple polypeptides coming together.
And that could be in one protein or in a quaternary structure.
Okay, so, sorry, the question, could that be in one protein or in a quaternary structure? So the protein has a quaternary structure. So for instance, hemoglobin. Hemoglobin people talk about as a protein. It's got 4 polypeptides that are part of it, 2 alphas and 2 betas. So the tertiary structure is always going to be of one polypeptide, yes. Yeah, Dan. Yeah.
Again, on the chart, would you see a domain potentially in the quaternary structure? Would you see it in any of the other 3 up there?
Okay, so the question basically is, well, again, we're getting back to this whole domain question here again. But the domain is going to be a separately folded, and we'll get to folded in just a second piece here, of a polypeptide. So you're not going to talk about a domain here in quaternary structure. You'll talk about domains in tertiary structure. This tertiary structure here looks like it's only one sort of structure. And so that would be a single domain here in that particular polypeptide. Confusion, less confusion, more confusion? Okay, so let's talk a little bit about folding. First alpha helices and beta pleated sheets, but then talk about folding. I think I've kind of talked these to a little bit to death now. These secondary structures are all about interactions with the backbones. The backbone atoms are forming hydrogen bonds, giving you an alpha helix or a beta pleated sheet. Again, the important things about these, as well as that, are that the individual side chains, which are going to be important for forming those tertiary structures, are going to be sticking out. But just because of the nature of this process, all of the R groups here are sticking out. And if you count 7 of these R groups, 1, 2, 3, 4, 5, 6, 7, as you know, I missed one here, 5, 6, 7, you're going to end up on the same side of this alpha helix. 1, 2, 3, 4, 5, 6, 7. Now, obviously, this has not been done terribly well done. It's not in 3 dimensions. But here, this R group is on the same side of your alpha helix as this R group. And what that means is that anytime you have the same amino acid, every 7 in your primary sequence, it's going to be on the same side of your alpha helix. Mostly important for what? Leucine zippers, exactly. Beta pleated sheets. Here, again, these are hydrogen bonds formed between backbone atoms, sticking side chains away from that particular secondary structure, again, so they can form tertiary structures, and in some cases also the quaternary structures. In a tertiary structure, you may have some amino acids that are sticking out of that tertiary structure that may be very important for forming quaternary structure. So how do you get these? again, it's really about the weak interactions, hydrogen bonds, ionic interactions, and hydrophobic interactions. One thing that we didn't talk about, I forgot to mention, there's one particular amino acid which is really weird relative to all the other ones. That's cysteine. Cysteine can form what are called disulfide bonds. This actually can form covalent bonds between different secondary structures in order to give you more constraints on your particular protein structure. So disulfide bonds, again, as always, an exception to what I've been talking about before. These are covalent interactions which are important for getting a tertiary structure, and sometimes even a quaternary structure. These disulfide bonds, two cysteines, can be between two different polypeptides and giving you a quaternary structure. Okay, hopefully this has become clearer now. Multimers, multiple polypeptides coming together into a protein. You'll have dimers like this. This is the CRO, and here's a nice example of where we have secondary structures and the side chains on secondary structures interacting with each other in order to give you this multimer. Here's a tetramer, 4 different polypeptides. Yeah, Diana.
For multimers, do the individual polypeptides have to be the same to come together into multimer, or is it different?
So the question here is, do they have to be the same or can they be different? And they can be both. They can be the same or they can be different. And we'll talk about this more a little bit later on, but it will be a homomultimer where they're the same or a heteromultimer where they're different. But you can definitely have both. Both of these are cases of homomultimers. So a homodimer, 2 of the same sequence polypeptide. Here a homotetramer, 4 of the same. Hemoglobin, which sort of mentioned before, it's also mentioned quite a bit in the textbook, is a heteromultimer. It's got 2 alphas and 2 betas. What about the core of a histone? What would that be? What kind of multimer? Is that going to be a homomultimer or heteromultimer? Heteromultimer, because you've got 4 different proteins in there. Okay, and then the huge, biggest one that we've talked about so far, protein, and people will just talk about this as a protein. The DNA-dependent RNA polymerase is a protein that has, depending on the organism, between 12 and 14 polypeptides that come together to make one protein. So this would be what kind of multimer? Homomultimer, heteromultimer? Heteromultimer, clearly extremely heteromultimer. And so these are all of these things that are coming together here. Allosteric regulation, okay, really briefly, this again is probably more important in a biochemistry course, but we'll talk about some examples here, particularly in terms of some of the G proteins, etc. The basic idea here is that normally with a particular protein, specifically if you've got enzymes, there's one little part of the protein that's important for its activity. And the rest of the protein is basically just there to hold that little part in the right place. Exceptions to that are allosterically regulated proteins, and allosteric just means somewhere else in the structure. So this somewhere else in the structure is also going to bind to something. And we can either have allosteric activators, which bind to part of the molecule and activate somewhere further away, or negative allosteric regulators that will bind and block activity. The whole idea here is you have some interaction in one part of the protein that causes a difference in the structure in some other part of your protein. It's a very important concept. So we talked a little bit about this in terms of the major signaling molecules, or signaling proteins, I should say, that you find inside of the cell. And this is a signaling pathway, really. Those 2 are either phosphorylation or so-called G proteins, GTPaces, GTP binding proteins. All those 3 things mean the same. So G protein, GTPase, and GTP binding proteins. So all of those are the same. That's on this side. That's a slightly more complicated situation that we'll talk about in just a second here. For protein phosphorylation, you have a protein kinase. All that kinases do is they put phosphates onto whatever this happens to be. There are also nucleotide kinases that will put phosphates onto nucleotides. So this protein kinase takes the gamma phosphate, the last phosphate in ATP, and through coupling the hydrolysis of that phosphate, makes a covalent bond here to this protein, and now changes the structure of that protein in one particular place. Sometimes that'll be an allosteric place. Sometimes it'll be directly in terms of what kind of signal is coming off of this. This covalent bond of the protein is then removed by a protein phosphatase. Phosphatase taking the phosphate off. So here, you absolutely require protein kinase. You absolutely require protein phosphatase. Since it's a covalent change, changing that particular part of your protein. On the other hand, G proteins, GTP binding protein, GTPaces, do not absolutely need extra proteins. In the cell, they usually do, because otherwise these reactions would be extremely slow. But they don't absolutely require these, because a GTP binding protein, not surprisingly, will bind to GTP. But this is not a covalent attachment. It's a non-covalent attachment, so lots of individual non-covalent bonds. It will bind to GDP, that's G with two P's here, or GTP that we have down here. This GTP-bound form causes a conformational change and a signal. At some point, there will be hydrolysis of this high-energy bond right here. So this phosphate's not a high-energy bond. Here we have a high-energy bond because it's a triphosphate. This bond is hydrolyzed, and you have GDP-bound form. Eventually, and this can happen, again, spontaneously, this GDP will be released, GTP will be bound, and this whole cycle continue just with the G protein. In the cell, all of this is very slow, particularly this step. So you need in the cell a guanosine nucleotide exchange factor that takes off the GDP, puts on GTP, and thereby activates this process. Hydrolysis often is stimulated by a GTPase activating protein or gap protein. Again, this is not necessary for the process. It just speeds it up, and particularly important, again, for this purpose, for molecular biology, it allows it to be regulated. So you can turn up or turn down this particular process. Okay, any questions on DGPAces and kinases, et cetera?
Just for clarity,
yeah.
the GTP was non-covalent bond, but the phosphate is covalent bond?
Yeah, so here, just basically repeat what I just said. The GTP in a GTPase, GTP binding protein, et cetera, is not covalently attached. It's multiple non-covalent interactions. Whereas the phosphate that gets put on by the kinase is covalently attached. It's covalently attached to serine, threonine, or tyrosine in the case of eukaryotic systems. Different in bacteria. But as far as we're concerned, serine, threonine, and tyrosine are those that have a phosphate covalently attached to them. Yes?
I'm confused because you said the kinase adds the phosphorus, yet I thought kinases hydrolyze, which is catabolic.
Okay, so the question here really has to do with kinases versus phosphatases. So usually, again, 90% of the time, ACE means that that is going to be hydrolyzing something. That's different in this case. Well, it is hydrolyzing the phosphate off of ATP, but it's then forming the covalent bond onto the protein here. So kinases are always adding phosphates, or better way of putting it is transferring the phosphate from a nucleotide triphosphate to whatever its substrate is. Protein kinases are going to phosphorylate proteins. Nucleotide kinases are going to phosphorylate nucleotides. Lipid kinases are going to be putting a phosphate group on lipids. Carbohydrate kinases are going to be putting phosphate groups on carbohydrates. Yeah?
Just to clarify, so the G protein doesn't need any enzyme, extra enzyme to just function of its own, while the protein kinase needs two enzymes. So it's 2 versus 0.
Yeah, so again, this, yeah, basically, were someone to ask a question on an exam, how many enzymes are absolutely required for G proteins versus, and it's really not a protein kinase per se, it's the process of protein phosphorylation here. You do need to have 2 enzymes for the whole process, putting a phosphate on and taking the phosphate off, whereas for G protein binding, you don't absolutely require, whereas in the cell, usually you'll have these other proteins that are involved, but they're not enzymes. The nucleotide exchange factor is not catalyzing any reaction. It's not making or breaking covalent bonds. The DGPase activating protein is not making or breaking covalent bonds. It's the actual protein itself, the DGPase, which does that. Yeah?
So the GTP binding protein, so the GAP and the JEP are not enzymes. They're regulatory proteins?
Yeah. So the GAP and the GF are regulatory proteins. They're not enzymes. Okay, hopefully this is clear. Now, Devin's still got a question. Yeah.
So these are allosteric processes.
You can think of these as allosteric processes. It's probably not the best analogy, because usually with allosteric, there's a separate molecule that's involved in binding. So these are actions at a distance. I wouldn't necessarily say that they're in a classic allosteric case. Yeah? Ah, okay. What's the distinction between an enzyme and a protein? Just use that as an example. So here, an enzyme is going to catalyze a particular reaction. And all the reactions we're going to talk about here in terms of enzymes are going to be breaking or forming covalent bonds. And there are plenty of proteins, for instance, the substrate of the protein kinase, that may or may not have enzymatic activity. And we'll talk about lots of DNA binding proteins. What's their function? Their function is to bind to DNA. Histones, great example. Those are not enzymes in any way. They don't catalyze any reaction. They bind to DNA. So the important thing there is really the chemical bond formation, covalent bond formation or breakage. Hopefully that's a little clearer. So people do use, hopefully not me, enzymes and proteins interchangeably. But there are lots of structural proteins, et cetera, that have no enzymatic activity whatsoever. Okay, finally talk about nucleotides. Woohoo. So I just briefly wanted to talk about this in terms of discovery of DNA being the genetic material. I think that this was pretty definitive proof, but people only believed it when they saw the viruses later on with Hershey and Chase, that you had a transforming principle that was degraded by DNase and not other things. Important here that I forgot to mention the first time around is that this process that Avery did, this transforming material, really depends on a particular cell, in this case, the rough cells, these guys right here, picking up a piece of DNA from the environment. You just put those two things together, I should say. A piece of DNA together with this cell. This cell picks up DNA from the environment and incorporates it into its genome and transforms in this particular process. What would you call this in terms of gene transfer, new gene formation? Horizontal gene transfer, exactly. And horizontal gene transfer, this case is called transformation. And it turns out that lots of cells, like E. coli, unfortunately, this wonderful model organism, it's actually really hard to transform. And these particular organisms, these are they've changed multiple names over the years, so I won't get into it. But these are what are called naturally competent or naturally transformable cells. And so this only works because you have this particular mechanism for picking up DNA from the environment and doing horizontal gene transfer. So that's the whole idea of the transforming principle. We talked about Hershey-Chase. Hopefully this is very straightforward. I wanted to emphasize here that this is actually the overview of two different experiments. So there's one experiment that was done with labeling with P32. P32 labels phosphates. There are lots of phosphates in our DNA. So you've mostly labeled it with this radioactive phosphate. In a completely separate experiment, you can label with S35. You won't find any sulfur in DNA. You find some sulfur in proteins. These are amino acid side chains. And you do an experiment, just a second, I'll get right back to you. If you do one experiment where you have labeled P32, break off the leftover viral shells, that P32 is transferred. If you do an experiment where you have S35, break off the S35, you have no radioactivity that's being transferred. Yeah?
In the example of like the naturally competent cells, are the sequences of DNA like being facilitated into the bacterial genome, or are they like transposable elements and sort of self-inserting?
So the question here is getting at, I'll paraphrase if it's okay. How does transformation work and how do these things get incorporated into the genome? We'll talk about homologous recombination after the midterm. But it is mostly how that happens. Nucleotides. Everyone says, am I going to have to draw a nucleotide on the exam? Previous years, yes. That was when there were about a third of you taking my class, and I could actually grade these things appropriately. There will not be a draw a nucleotide question on the exam. Almost certainly there will be a structure to look at and decide what's wrong with it, what's right with it, what's the base pair, etc. So it's great practice drawing out these nucleotides for individual exams. 5' and 3', hopefully we're doing this to death, but it's very important because we're continuing to talk about how polymerases happen, etc. The 5' position and the 3' positions are on our deoxyribose, which is a 2' deoxyribose in DNA. It's a 5' phosphate, 3' OH. And then the base is always attached here at the 1' position, also known as the glycosidic bond. These bases, 4 different bases, 2 purines, 2 pyrimidines. The way I remember this, purine, short name, big molecule, pyrimidine, long name, small molecule, as far as this is concerned. How do they fit together? Hydrogen bond interactions between the individual bases, but also very important is that these glycosidic bonds are in the same place in space, whether you have a GC, an AT, a CG, or a TA. And this was, as far as I'm concerned, the real major breakthrough that Watson and Crick managed to do in coming up with their model for the structure, is that it doesn't matter what the base pair is, it still gives you the same structure. So that's the big, hopefully, take-home message here. Many people have asked, is this the kind of base pair which you'd want to see on an exam? It's actually missing a bunch of things. It's missing hydrogen atoms. It's missing the bases. It's missing the phosphates. So people have written this up on an exam and not gotten very many points. How do these come together? Again, it's the double helix model built by actually the machine shop at Cambridge, then put together by Watson and Crick. Anti-parallel strands. Again, anti-parallel was something that they had no idea was the case from the other data. It's the model only worked that way. Yeah, sorry, your question.
Going back, so you say the structure is incorrect on the slide, so how are you supposed to find the right structure to learn?
You look and see what the structures of the individual nucleotides are. Almost all of these are going to be simplifications. So the question is, if this isn't the right one, what are we supposed to learn from? You're supposed to find the individual, don't cackle too much, is find the structure of the individual nucleotides and put them all together. So here, for instance, we've got these individual nucleotides. There's a full nucleotide up here, but interestingly enough, these guys have H's hanging off of them. This one doesn't. Why not? Because they were lazy. And I don't expect you to be lazy. So you're going to have to put these things together. That's kind of the point. So make sure that they're complete. If you got any questions about it, scan them and email them to me. Yeah?
So the ideal form and structure is what it's showing for the period on these slides, basically. Just to know to study from if I was to draw it and check myself.
If you were to be drawing something and checking yourself, make sure that you include all of the hydrogen atoms on here and all of the bases, which here, you know, look, just got an O hanging out here. There's clearly a bunch of stuff missing here, right? So if you look carefully at some of the old exams that have asked you to draw base pairs, they will say, include all of the hydrogen atoms, include the polarity, include major and minor grooves. Yeah, there's a question in the back. Oh.
Is it implicit, like a lot of things online? I heard that they don't actually always include carbon and hydrogen in the drawings. I mean, you're asking us to, but...
Yeah, so, and lots of examples, they do not include them, but I think it's important that they're there. So I would study them with all of these things in there. Because someone may ask you a question, like here. So this would be an example. Here, what are the bases missing? What are they missing? Hydrogens. There should be an extra hydrogen hanging off of here. There should be a hydrogen down here. There should be a hydrogen there. There should be a hydrogen there. So that's the answer to your question right there. If I were to ask that. Which I probably won't since I'm asking it now. Doesn't mean you can forget it. So getting back to our double helix, here, if we don't get through the rest of the stuff today, we still talked about it in lecture, so it's still fair game for the exam. So major groove, minor groove, again, this just comes from the model. You've got pieces of your DNA which are going to have the variable kinds of the atoms. You have a nucleotide, which are going to be up here. For instance, the major groove would be up here. Minor groove would be down here. And this is great to have sequence-specific interactions, which we'll talk about as nauseum after the exam. They're going to be happening in the major groove. Probably through evolution, the amino acids that stick out of an alpha helix are exactly in the right place to interact with the bases in the major groove of DNA. Oh, by the way, no one has told me what the major problems are with this model up here. So just something to think about. The double-stranded DNA structure, I mentioned that you have very sharp transitions when you heat up a DNA. So normally, the base pair stacking is stabilizing your DNA up to a certain point, and then as soon as some of the pieces start to come apart, the rest of your DNA will come apart very rapidly. So that I mentioned. What I should have emphasized is that this process is completely reversible. So if you heat up a DNA to a high temperature and the two strands come apart, the optical density increases, you can cool this back down and end up with this double-stranded DNA where all of those bases are matching exactly. And this is important because when we talked about those karyotypes, where we've got the individual fluorescent tags in any given chromosome that will be different, that has to be because you've heated up the DNA, cooled it back down with these short pieces of DNA, which will bind to each of those individual chromosomes. So important here is denaturation and renaturation. Denaturation, getting those two strands apart. Renaturation, getting them to come back together. Big picture with human chromosomes, we've got a bunch of non-coding DNA in them. That's basically all that this was supposed to show you. And genes are relatively infrequent, even in chromosomes that are very gene dense, like chromosome 22. How many genes do you have in the human genome? Asking like about do I expect us to know delta G's for any given reaction? Do I expect you to know the absolute number of protein coding genes in the human genome? No. Do I expect you to know it's about 20,000? Yes. That there are a whole bunch of non-coding RNA genes, and we really have no clue what that actual number is also, I think, something which is reasonable to mention. That the exons themselves are usually around, it's just not even in here, 150 base pairs. Why? Because that's the size in one particular nucleosome. I think those are reasonable numbers to remember. Some of these other ones I don't think are that reasonable. Speaking of nucleosomes, how do we know that it's 150 base pairs? If we take nucleosome-bound DNA, cut it with an endonuclease. So we talked about exonucleases that chew in at ends. Endonucleases cut in the middle of DNA. So they're not dependent on the ends. Got with an endonuclease that will cut DNA that's not being protected by proteins. Then you end up with about an 150 base pair piece, which you can see here. And then some pieces were not completely cut, some pieces less so as well. So you've got one nucleosome, two nucleosomes, three nucleosomes, four nucleosomes, cut to multiples of 150 nucleotides with this endonuclease. If you now purify the proteins that are present in this DNA, you'll find the vast majority of them are the heteromultimer of H2A, H2B, H3, and H4, dimers of each of those. Those fit together in the nucleosome, wrapped around almost 2 times with H3, H4, H2A, and H2B with these tails hanging out. The histone fold, these 3 alpha helices are practically identical for each of these histones. The tails are very different, and of course the tails are where you have lots of modification is going on. Sequence specific versus non-sequence specific. I answered a question about this probably rather late last night online, but the basic message here is that the vast majority of nucleosomes and the interactions with DNA are non-sequence specific. That's because most of those interactions on the slide that I'm not showing here, where you have hydrogen bond interactions, you have charge-charge interactions, most of those are with the backbone, sometimes a little bit in the minor groove of the DNA. So these are non-sequence specific interactions. However, if you're trying to do an experiment where you want to know a nucleosome is in one particular position, you can artificially design a stretch of nucleotides that every 10 nucleotides you've got AT base pairs. And what that does is it allows the DNA to bend. And it bends a little bit more easily than a piece of DNA that would have random A's, T's, G's, and C's. And so if you've got this designed piece, or maybe an evolutionary conserved piece of DNA that has those particular nucleotides at different places, nucleosomes are going to bind more preferentially to that because the DNA is easier to bend there. But that's going to be relatively rare, and usually these are going to be non-sequence specific interactions. So if you're doing an experiment where you want to have nucleosomes at a very specific position, design a piece of DNA that's got AT bases every 10 nucleotides. And that way you'll have much more specific interactions there. Even when you have these specific interactions, the nucleosomes and the the DNA relative to the histones is coming on and off all the time. It's a very dynamic system. And hopefully in some of these videos you see everything moving around, bouncing around, etc. This then allows sequence-specific binding proteins to interact with DNA even if it's in the middle of a nucleosome. Turns out this is not enough. The reason we know that is because there are chromatin remodeling complexes. Chromatin remodeling complexes bind to nucleosomes on DNA and mess around with it. How do they mess around with it? We talked about the DNA moving relative to the nucleosome, so just pushing and pulling the DNA relative to the nucleosome. That's one of the things that these chromatin remodeling complexes do. They also will exchange some of the histones for other histones. They can also completely dissociate the histones from the nucleosome. There's usually another enzyme, or for that matter, just the histones, which will associate with this DNA that a chromatin remodeling complex has dissociated. And again, I discussed this on the discussion part of D2L late last night. So if you're interested in more details on that, take a look there. The histone tails, these are the non-conserved part of the histones, the parts that are not in the histone core. These then get covalently modified. Covalently modified means they've got enzymes which are being used to make these covalent modifications. So histone acetyltransferase, histone methyltransferase, enzymatic changing, making covalent bonds. The most common of these are the acetyl groups and the methyl groups. Most of these are happening on lysines that are very common in these tails. Not surprisingly, positively charged, interacting with negative charges in your DNA. But it's not just charge-charge interactions. These modified histones interact with histone reader complexes. Histone reader complexes, again, are important for interacting with these specific atoms and the specific modifications that are happening on these histone tails. Histone readers, just by themselves, are going to bind to and recognize these modified histones. That's all that a histone reader does, is it binds to these modified histones. Most of these histone reader complexes will also interact with enzymes. So, histone reader, enzyme, not an enzyme. Not an enzyme. Interacts with enzymes that can then modify more histones. So usually the first step in one of these changing of the chromatin structure around where you have a histone modification is going to be binding of a histone reader and then changes to histones nearby. So this would be the histone writer complex. Yeah?
Are the code readers considered sequence-specific or non-sequence-specific?
So the question is, are code readers considered sequence-specific or non-sequence-specific? The code readers are modification-specific. So they're not sequence-specific. They're modification-specific. They're going to just bind to histone tails that have been modified. So they're not sequence-specific. To get, this is the chicken and egg question here. How do you get that histone modification in the first place? Then you'll have a sequence-specific DNA binding protein that can associate with one of the enzymes to start the process. Okay, are we at 10:05 yet? Okay, just basically this is what we talked about already. You have the sequence-specific binding protein that will associate with one of these enzymes, which will modify the code reader, this green thing, which will allow this to propagate. You have to stop this propagation at some point. That is due to the barrier proteins. I just wanted to finish up this here last one, condensins. I mentioned right at the beginning today there was one slide that I changed. That's because it was unclear the first time that I explained it. Someone sent me an email, said, but the video doesn't agree with your lecture. They were right. The video was right. It doesn't agree with my lecture. So my lecture was probably not completely right here. So here, condensins 1 and condensin 2. Condensin 2 is the one that forms most of the helical structure in the middle of metaphase chromosomes. Also stacks the loops on top of each other. Condensin 1 interacts with condensin 2, so it also ends up forming a helix, but condensin 2 is the first one, and then helps compact these individual loops, so pulling the loops together. So you've got loops and helices here. Good luck on the exam on Wednesday. More questions, come grab me now. Send me emails. I will be trying to answer them up through Wednesday morning. And good luck.